called passive replication of bacteriophage. The same figure or the same event is observed in form of double-stranded DNA, where we can even observe the joining, then the cutting, and the basic recombination, means the optional genes will attach with each other and they form the lambda prophage structure, as you can see on the screen. Now, the important events which can affect the stability of lysogeny is the induction process. So, what is induction? So, all we know, the prophage, it is a lysogenic state of the bacterial host with the bacteriophage genome. So now induction is the one type of effect when any stressed condition, or we can say the exposure of UV cells to the prophage, means if the prophage is there and it is exposed to the UV light, then it will cause once again the recircularization of that bacteriophage genome from the host cell, means it will get separated from that host cells, and once it is circularized, it will start or it will take up the whole control from the host cell and starts its own replication, assembly and multiplication of cells. And once the number of cells are formed, they will break up the cell wall of the host and they will cause the release, means the lysis. So induction is ultimately the process of conversion of prophage form or the lysogenic state of the bacteria into the lytic state of that bacteria. Now, so overall, excision, lytic growth, progeny formation, and ultimately lysis. So as you can see on the screen, this is shown in terms of genetic evidences. So first we are observing the repressor fragment of prophage, then it is exposed. Once it is exposed with UV light, the circularization of that genome take place, and once it is circularizing, the enzyme excisionase cuts it from the host genome, and that host genome will get separated, and once again the free form of bacteriophage will take up all the control from such bacteriophage and starts its own replication, which will ultimately leads to the formation of progeny and causes the lysis of the host cells. So these are the typical steps of induction, excision, lytic growth, progeny formation and lysis. Another important event which is related with lysogeny is called immunity. So what is immunity? So as you can see on the screen, this is the structure of prophage. So once it is, if suppose it is infected with a bacteriophage and other lambdaphage comes from outside, then it is not capable of causing any infection to the already infected host cells. Means if one bacterium cell is already infected with lambdaphage, then other lambdaphage, similar type of other lambdaphage cannot infect the same host and it cannot get replicated or infected. So this phenomenon is known as the immunity of that host cells from the infection by second or other type of bacteriophages. Now, though our main topic of today's discussion is the regulation of lysogeny, so we must have some basic idea regarding the overall genetic structure of these bacteriophages. So as you can see on the screen, this figure shows the overall gene organization, or we can say the genetic map of typical lambda bacteriophages. So at the bottom there are the genes, structural genes, which are mainly about the head and tail genes. Then on the left you can see the genes of recombination. Same way on the right hand side you can observe the genes which are responsible for the replication, and in between these genes of recombination and replications you can find the various gene region like the C1, C2, N, CRO, Q. These are known as the regulatory region. And near the replication region also you can find the region of genes which is responsible to cause lysis. So all this region, now we are going to see all these regions in a linear form. So this is the same figure which is shown in a linear form and which shows how these typical genes are arranged. So basically overall the phage genome are 30 genes and their length or the size is around 48,502 base pair long, and that can actually cause the life cycle of a typical bacterial phage. Now we'll see some of the details how these various groups of genes work. So first we'll start with the genes which are called the head and tail genes. Means this code for the structural proteins of the bacteriophage capsid as well as the terminase enzyme required to process rolling circle multimers into the unit genome length pieces during packaging. Means the head and tail formation as well as its packaging material. Then these are some of the genes, means the examples of various genes which involved into the head and tail assembly. The next, the important group is known as the recombination genes, so mainly which codes for the INT or XIS, which are required for either INT for integration of the bacteriophage into the bacterial host chromosome during the lysogenic growth, and XIS is responsible for the excision from the bacterial host chromosome during that induction process. Some of the examples of such recombinant or the genes required for recombination are as shown in the screen. The next important group, which will be later discussed in more detail, is the group of regulation region. So this includes the immunity region as well as the genes that are responsible for the controlling the switch between the lysogenic and lytic growth. Means the important is that Q anti-terminator protein, which is produced or it is the product of Q gene, as well as the anti-qRNA and PR dash constitute a second regulation region. So these are the basic, some of the important examples of regulatory genes which you can observe on the screen, like the N, which mainly affects on the early gene transcription, then Rex B, Rex A, and most important one are the C1, CRO, C2 and REN. These are some of the genes which are having no definite role during the regulation. They are observed in the regulatory region, but their role, their definite role, is still unknown. And ultimately, the replication region mainly includes just two replication products, O and P, that is the origin of the replication. And finally, there are four genes in the lysis region which are responsible to cause the host lysis, that are ORF64, S, R and RZ genes. So these are the basic composition and the gene organization of the typical lambda bacteriophages. Though our main topic of concern is the lysogeny and its regulation, now that will be further discussed by my colleague Mr. Yasin Mansouri, and he will continue this topic or discussion. Thank you very much.
Good morning, viewers. It's a pleasure to be here to be part of the whole event of BISAC, and I take this opportunity to thank all the staff involved, all the persons involved in achieving this great adventure and this great target of teaching the students all across the Gujarat in this beautiful program. Now let me continue my topic of discussion, our topic of discussion, that is the regulation of lysogeny. As my colleague discussed all those genes, this is just a tabular form of the genes starting from N, which is an N-determination protein. The whole transcription begins with the role of the N and the CRO, which is listed below. Apart from this, there are O genes, the P genes, which are initiation of the lambda DNA replication, the Q gene, which is again an N-determination protein, the C1 molecule, the most important molecule of regulation, that is C1 repressor, it's a repressor protein. The C2 and C3 are helping C1 to work, and they are activators and stabilizers respectively. The CRO, which is a protein which inhibits C1 synthesis, the GAM, which is required for the rolling circle replication, the red gene, which is involved in the recombination, the integrase gene, as my colleague previously mentioned, is the gene for the integration, the formation of lysogeny, and the access gene, which is for the conversion of the lysogeny to the lytic, that is the accessase enzyme. Now, as we can see further, the same map including the most important genes that are part of this regulation, that is the integrase, the C3, the N, C1. Among these, the three genes that are specifically required for controlling the lysogenic pathway are the lambda C1 gene, C2, and the C3 gene. So the whole network begins with the early gene expression, in which the promoter left and the promoter right are transcribed, and this transcription leads to the expression of the N and the CRO. So as you can see in this flow, that promoter left and promoter right are transcribing the N gene and the CRO gene. Now this N gene and the CRO gene transcription begins the transcription and determination function by the N. So this leads to the expression of the C3, C2 and the Q genes. So as you can see here, the N has transcribed itself into C2, C3 and the Q gene. Now, additional regulatory steps of this network is that the N is auto-regulated by a negative feedback loop, some sort of feedback inhibition. The CRO represses expression from the PL and PR promoters, and the same CRO is also negatively repressed by a negative feedback loop. Now, as you can see here, the negative feedback loop of N and the CRO are visible in this slide. Now, the lysogenic pathway begins with the function of the C2 and C3. The function of C3 is to stabilize the rapidly degrading C2. The C2 is actually getting degraded by HF1 protease, but the C3 stabilizes this degradation, and thus the C2 gets activated and it activates the product, that is the integrase enzyme. The other function of C2 is to activate the C1 synthesis. The C1 repressor, that is the C1 protein, it represses the promoter left and the promoter right, and C1 auto-regulates its expression from promoter repressor maintenance, that is PM or PRM. As you can see these steps here, that is the functioning of the C3 and the C2 with the blue lines on PL and PE and integrase, and apart from that PE, that is promoter establishment. It is promoter repressor establishment, PE, which has a function on the C1, which is visible on the left corner of the screen. Now, the C1, because it is a very important molecule, it is further discussed that is C1 repressor, the protein, the C1 repressor protein is made up of two monomers, that is, it is a dimer which is connected by a connector molecule. So the C1 repressor is made up of 27 kilodaltons molecular weight. It has two distinct domains, that is the N-terminal domain, which is having a residue from 1 to 92, which provides the operator binding site. The second monomer, that is a C-terminal domain, it is from the 132 to 236 amino acids, and it is responsible for the dimerization. These two molecules are connected by a connector molecule which is having a 40 residue. So when this repressor is digested by any protease or by a protease, each domains are released as a separate fragment, that is the C and the N. Now the second important molecule of this whole story is the CRO. The structure of the CRO is that CRO is a dimer, that is, it is a monomer having 66 amino acids that are linked. This is the structure of the CRO. Now the main point, that is regulation. The regulation begins with, as I mentioned earlier, the C2 and the C3. The gene product of C2 and the C3, they bind with the PRE and activate PRE. PRE is the promoter for repressor establishment. So this binding with the promoter repressor establishment has an effect on the C1 gene. That is, the activated PRE facilitates the transcription of the C1 gene, and thus C1 repressor is produced, the central molecule of the whole phenomenon. Now this C1 repressor has two functions. First, it binds with the PRM. PRM is the promoter for repressor maintenance, and thus it activates this promoter. So the PRM ensures the continuous synthesis of C1 repressor, as you can see in the slide, in the flow. The second function of the C1 repressor is that it binds with the operator promoter left and operator promoter right, and thus it inactivates both these promoters. So this will stop the transcription of phage structural genes. That is, the transcription of the phage structural genes will be inhibited. So the C1 repressor does not allow the replication of the lambda phage DNA to occur. This favors the lysogenization. So that is how the C1 repressor has a very profound effect on the lysogenization. Now, once this lysogenization is induced by some UV rays, there comes the role of the second molecule, important molecule of CRO. Now, for the transcription of the phage structural genes, the CRO gene product is required. GP stands for gene product. So this CRO protein, it binds with the